So it's my pleasure to introduce you, Dr. Hudson. You're a rheumatologist, of course, and epidemiologist at the Jewish General Hospital and Lady Davis Institute, and an associate professor and member of the Division of Experimental Medicine in the Department of Medicine at McGill. Dr. Hudson pursues research in systemic autoimmune rheumatic diseases and is a founding member of the Canadian Scleroderma Research Group, also known to us as CSRG, which since 2003 has involved 15 sites across Canada, 20 clinicians, and 10 basic scientists. They've secured over $4 million in funds, trained over 30 graduate and postdoctoral students, contributed to over 100 peer-reviewed papers, and become an internationally recognized scleroderma research program. Dr. Hudson has worked on a broad range of projects and contributed to landmark papers. In addition, given the rarity of the disease at hand, speaking of scleroderma, of course, she has also established several international collaborations with leading scleroderma experts worldwide. She currently co-leads the Scleroderma Clinical Trials Consortium working group on scleroderma renal crisis, and has recently developed new interest in epigenetic signatures and cellular therapies for scleroderma. So a long list of accomplishments there. We're so lucky to have you, and looking forward to your research updates today. Thank you for being with us, and I will give you the floor.
Thank you very much. Thank you for inviting me. It's really an honor for me to be here. Before I start, I'm going to figure out how to share my screen. Here we go. I hope you're seeing this.
We are, yes.
Excellent.
Yep,
okay, very good. So it's a difficult time, I would say, to be presenting to an empty office like this with no feedback. So I hope that you can write in your questions, and I'll try to address some questions as much as possible towards the end of the presentation, and try to make this as interactive as possible given the circumstances. So I've been asked to address research, and I thought this was a real opportunity to talk to you about the Canadian Scleroderma Research Group, the past, the present, and the future, to show you a little bit of what we've been doing in the past and where we hope to be going in the future, and hopefully give you some hope. So my objectives here, I will speak briefly, describe the CSRG, so the Canadian Scleroderma Research Group, for those of you who are not familiar with it. And then I will break up my presentation in two parts. I'm going to talk about what I call phase one of the CSRG, which is perhaps the last 15 years or so, where we've been doing mostly epidemiological research, and I'll go over some of the stuff that we've done. And then in the latter part of my presentation, I'll go into what I now call phase two, where we're now trying to delve a little bit more into the mechanisms of disease, and hopefully some new interventions to help treat the disease. So for those of you who are not familiar, the CSRG was created sometime in 2003. The cohort was established officially in 2004. And we've been lucky over the last 16 years now to have participation really right across the country. It's been really a national effort. And I think it's part of this Canadian collaborative feeling has really helped to make a success of this research group. We've involved many sites, many investigators. We've recruited what we think is about 15% of all scleroderma patients in the Canadian population, which is quite a feat. We've had a lot of success with funding, and we've put out a lot of papers. So basically, that's who we are. So let me walk you through some of the epidemiological research that we've done. And for people who are less familiar, epidemiology, and when we talk about epidemiology, we talk about the health of populations. So in general, when we do epidemiology, we describe the population of scleroderma patients, what their health is, and what their health outcomes are. So that's what I'm really describing as a group. What does scleroderma look like? And the word that I just used, it's very descriptive research. So we describe what the problem is. So over the past few years, as I mentioned, we've published well over 130 papers, and I'm not going to walk you through all of those papers. I'm just going to highlight some of the more important papers that we've put out there. So of course, scleroderma is a skin disease, so we've put out several papers on skin, but a lot of people are doing research on skin. So we've tried to look where other people have not looked. And so we've tried to describe the subset of patients who have scleroderma without having skin disease. So that's scleroderma, cine scleroderma. We've also recently, or well, that wasn't so recently, about 5 years ago, we put out a paper saying that, there's so much focus on the skin, and we tried to use the skin, limited and diffuse and so on, to predict the outcomes of patients. But we think that it's more than skin deep, and I'm sure many would agree that scleroderma is more than skin deep. And I think that a better way of trying to prognosticate around this disease is by combining skin and autoantibodies, and then making subsets of patients in those different groups, and then trying to use those newer subsets to try to understand the course of the disease better. So interestingly, this latter paper has recently been validated by a large UK group and another large European group. And so we were the first one to sort of suggest that, and then other groups have now come along and said that there's a good value in subsetting patients not only by the skin involvement, but by their autoantibodies as well. Skin is more than just the skin as well. We've worked on digital ulcers. We have a lot of difficulty even defining digital ulcers. So this was a paper that we put out there trying to come up with standardized definitions of ulcers. And the calcinosis, and I'm sure many of you struggle with calcinosis. We struggle with calcinosis for you as well. And so we're slowly trying to make some headway in terms of understanding what, what's happening and how can we better manage the calcinosis in scleroderma. Lung has been a particular interest to us. We've put out several papers on lung disease. I've just highlighted a few here. Some of the papers that I highlight here, some of the work that we've been trying to do as well is try to identify those really useful predictors of who's going to progress and who's not going to progress. Because if we had those predictors, we would then be able to say, okay, I need to treat you, and oh, I don't need to treat you. So we've done a lot of work in trying to identify biomarkers of progression of lung disease. I would say in the last year, one of our more impactful papers has been this particular one that was, this research project was done by a young rheumatologist at the Université de Montréal here in Montreal. And what she looked at, we tend to look at patients with lung disease when it's already somewhat advanced. And what we realize is at best we can stabilize the disease. So her question was, what if we tried to treat the disease when it was still mild, it wasn't so advanced? Would we have more of an impact in terms of controlling the progression of the disease? And indeed, that's exactly what she demonstrated. And I think you will have some talks on interstitial lung disease, so hopefully they'll go into that. But the idea here is perhaps we were waiting too long before initiating treatment in some of our patients, and perhaps we can preserve lung function if we introduce treatment earlier. And in fact, I will mention that this paper was highlighted as one of the top 10 research advances of 2019. So really starting to look at how can we have this window of opportunity, treat scleroderma earlier, and perhaps have better outcomes for our patients. So quite impactful research as far as I'm concerned. We've had also a young rheumatologist here in Montreal, Nicolas Richard, who's been particularly interested in the issues related to the gastrointestinal tract. There was very little literature related to fecal incontinence. He did perhaps the largest study on that. And he also looked at the associations with severe GI disease. And I highlight that here because I will subsequently tell you that this work has helped us conceive of newer projects and more interventional projects on how can we try to intervene on the GI disease in our patients. I have had a particular interest, as you heard, in the scleroderma renal crisis. There was a question of whether we could prevent scleroderma renal crisis using ACE inhibitors, and the hypothesis was that we should not do this. And in fact, this study that we did about 10 years ago supported the fact that we should not be using ACE inhibitors to prevent scleroderma renal crisis. And then subsequently, we've gotten together with international experts. You heard the Scleroderma Clinical Trials Consortium, trying to better understand what scleroderma renal crisis is. And we've had some papers in high-profile journals. And again, you'll hear about what this particular work has led to recently in a minute. Oh, well, there you are. So we've used our 10 years of experience of looking at scleroderma renal crisis to now launch an international study on scleroderma renal crisis. We started this in January of this year, and as of yesterday, when I asked my assistant, I think we already had recruited almost 60 cases and we have participants from just about all around the world. So I think that this will be very, very valuable data in terms of trying to understand what's happening with scleroderma renal crisis. Hopefully we'll get some results in the next year or two. We have been very fortunate to work with an international expert on autoantibodies by the name of Marvin Fritzler in Calgary. And so with Dr. Fritzler, we have really put out a whole lot of papers looking at autoantibodies in scleroderma, and not just the autoantibodies that we are familiar with, but trying to look at newer autoantibodies and seeing if that might help us make better diagnosis and make better prognosis. So that's been very exciting over the past few years to do this sort of discovery research with Dr. Fritzler. The other thing, and I'll speak about genes in a minute as well, scleroderma, I'm sure everybody is aware, is a relatively rare disease. And so it's hard to do genetic studies locally if you want. So we have been invited to be part of these international consortium to understand the genetics of the disease, and we've participated in several studies, what people often refer to GWAS studies, which are these large studies where we have thousands of patients, thousands of controls, and we try to identify some genetic markers of the disease. And we've been fortunate to be invited to participate in these projects. And finally, again, more in the descriptive world of things, we have we've worked very much with the Australian scleroderma group, who has also been very active in research. And so we were able to, using really advanced statistical methods, try to get a better handle of, what is the outcome, the hardest outcome yet of scleroderma is mortality. So we put out a nice paper measuring the mortality in scleroderma a couple of years ago. And we also, with this, the Australians, this was not available before in the field of scleroderma, but we developed an instrument or a tool to try to measure. It's one thing to measure mortality, but what about organ damage? And so this paper came out recently, and this is a really important step in research in scleroderma, and now being able to try to measure in a standardized way how the disease affects various organs. So this is just a snapshot of some of the projects that we've been involved with. And then I sit back and I ask myself, so what? So we've put out a lot of papers. What can we do with these papers? Why do we do this epidemiological research? And I think that there's many reasons why we do this research. One of the main reasons, or most obvious reasons, if you want, is in terms of advocacy. So I just mentioned that we measured in a very robust way what the mortality in the disease and the organ damage in the disease. And now we have numbers. We can actually, go out and advocate. This is, yes, a relatively rare disease, but it's a disease associated with high burden of disease. And so that allows advocacy groups to go to people who control health resources and so on to try to advocate for people with scleroderma. These are very, very valuable data to have in hand when it comes to advocacy. The other thing, one of the papers that we did quite early on is we measured health-related quality of life in scleroderma, because it's one thing to measure mortality, and it's one thing to say that the lungs are affected 50% and the kidneys are affected 25%. But I think it's also important to understand, and how do the patients live with all of this? And that's where health-related quality of life comes in. And what we found, in our research on quality of life, was that scleroderma patients live, on average, 1 standard deviation below the general population. So I say that and I say, well, what does that mean to live 1 standard deviation below the average population? So here, this is what a normal distribution looks like, okay? So the average population would be somewhere here. 1 standard deviation below or above the average accounts takes into account 70% of the population. So then there's 30% of people left, right? So 15% are going to have quality of life that's above 1 standard deviation above. but 15% of the population is going to have quality of life that's one standard deviation below the average. So what I'm really saying when I say that our patients are one standard deviation on average below the general population, it means that people with scleroderma are among the 15% of the population with the worst quality of life. So that's, that's impactful as well. So the mortality is elevated, the organs are affected, and the quality of life in this disease is really poor. So again, very valuable data to put numbers on this data to then turn around and try to go advocate for healthcare resources for our patients. And the last one of the other ways that we can advocate as well, and this is a paper we put out quite a few years ago, was in terms of estimating the cost. So, all in all, demonstrating the burden of this disease. The other thing that epidemiological or observational research can be useful for is it can then lead to interventional studies. So it's one thing to describe the problem, and then the question is, okay, so what do we do about the problem? And so based on some of the studies on the gastrointestinal tract that I mentioned, we went out and we designed a pragmatic trial trying to address problems of small intestinal bacterial overgrowth, and we were funded by the CIHR to do that. Similarly, I mentioned that Dr. Hoa looked at how did our patients do if we treated mild interstitial lung disease, and it looks like they do better. But this is just preliminary data. So now she's going out and she wants to test that specific hypothesis in a real trial. And so she's in the process of designing that interventional trial. And of course, I think throughout this meeting, you've heard about SPIN and all the fantastic things that SPIN has been doing in terms of designing interventions. Well, a lot of the rationale for the interventions that they decided to try to develop came from some earlier work from the CSRG demonstrating the impact of the disease on the psychosocial and rehab problems that are caused by scleroderma. So observational research can then lead to treatment research, if you want. I'm going to skip this because I think we're going to run out of time if I go through everything. And then I mentioned, all the work that we did in terms of autoantibodies and autoantibody discovery, and that can actually lead to new diagnostic tests. And so some of the data that we've generated has been used by some guys. I show one company here, but there are other companies out there who are very keen now to try to integrate these newer antibodies on scleroderma panels to give clinicians and eventually patients better tools in terms of trying to understand or make a diagnosis and understand how the disease is expected to evolve. The one thing that observational research does not do is cure the disease. And I think that people with scleroderma, they will support us in terms of trying to make the disease better, but what they really want is how can we cure this disease? And so that's why I see the CSRG, our phase 1 research has really been in terms of describing the disease. And now all of this massive information that we've accumulated, I think is now the impetus to go on to try to say, well, what can we really do? How can we help people in a concrete way with scleroderma? And so that's sort of the motivation between what I say the phase 2 of our research program, where we're now focusing more on some mechanisms of disease and trying to find novel treatments. And so that's what I'm going to address very quickly. So, for those of you listening who have scleroderma, you know that our treatments are very, very unsatisfactory, I would say. We have some treatments that will control the symptoms and that will slow down the progression of the disease, but I think we would all agree that we need better treatments. And if we don't have the treatments yet, we need to know what the targets are. How are we going to identify the molecules at the molecular level, what's going on that we can target to improve the disease? So here I'm going to share something personal with you, and I'm going to tell you that I'm an identical twin. And so my identical twin is actually a far better skier than I am, and she's a far better cook. And so despite the fact that we have exactly the same DNA code, there's got to be more than just the DNA, right? Because she's a better skier and she's a better cook. And the other thing, just to take it one step further, is we both have allergies, but interestingly, we don't have the same allergies. So I have allergies to, mild allergies to a few fruit or whatever, and she has very, very severe allergies to animals, horses, cats, and dogs. So for those of you out there who still think that your genes are your destiny, it's gotten much more complicated than that. I think that we now realize that the environment is a very, very important contributor to health and disease. And the way we are sort of now starting to integrate the component of environment in our understanding of health and disease is through science that's now called epigenetics. So for a long time, there's been an interest in the genetic code. So these are genes that are included here in the chromosomes. But over and above this genetic code, there are all these molecules, what I'm showing these little red tags here, and all these Ms here and there. That's over and above the DNA. So it is the epigenetic code. And so we're understanding, and we're coming to understand that some diseases caused not so much in the genes, but in the epigenetic code. And the other way that I explain this to patients is it can't all be about your genes, because you had the same genes you have now, that you had them at 25 years old, and yet at 25 years old, you didn't have this disease. So what is it? So it's not the genes that are completely responsible for the disease. There's got to be more to that. And we think that it's these molecules over and above the genetic code, so the epigenetic code, that are probably the ones that are being dysregulated, that are then causing disease. And what's really, really fascinating is that although your DNA, your genes, for the most part, do not change during your lifespan, your epigenetic code is highly, highly sensitive to the environment and can be affected by all sorts of environmental factors. And so we think that lifestyle factors, in particular smoking, diet, exercise, our gut microbiome can influence our epigenetic code, pollution, and the list goes on and on. So now it's starting to make sense that, okay, you have the same genes that you had when you were younger, didn't have the disease. Perhaps it's dysregulation at the epigenetic level now that has tipped you over into developing scleroderma. So this science has really gone ahead in the field of cancer. And so a few years ago, I thought, you know, the people in cancer are finding new causes of cancer through epigenetic dysregulation. And given that we know in our diseases like scleroderma, that there are probably some environmental influences, it makes a whole lot of sense to start looking at the epigenetic code of our patients. So at the molecular level, what's happening. So we've actually started to do this, and I'm certainly not going to belabor this point because it can get quite dry and complicated. But we have started to collect samples from our patients and sequence their epigenetic code. And we are starting to find really interesting things. So we're starting to find epigenetic dysregulation in genes that we already knew were involved in scleroderma, but we're also finding epigenetic dysregulation in new genes. And when you start to find signals in new genes, that gives you new targets to start to understand how is this contributing to the disease, and is this target druggable? Could we try to see if we can manipulate this target? If we can, because if you imagine what's fantastic about the epigenetic code is if you can tip it in one direction, you have to be able, it's reversible. So how do you tip it back into a healthy state? So first of all, what we're trying to identify is what are the genes that are dysregulated, and then try in the future, trying to see if we can put them back into a healthy sort of environment. So that's some of the mechanistic work that we've been doing. Now, in terms of treatment, I will talk about two types of treatment that we're starting to explore from a research point of view. And those treatments, you know, I'm sure you will hear talks on new drugs for lung disease and for other problems in scleroderma. But what's unique about the treatments that I'm going to talk about is that these are cells. These are human cells that can be used to fix the dysregulated immune system of people with scleroderma. And the two types of cells that are currently of interest in scleroderma are these pinkish cells, which is what we call hematopoietic stem cells, okay? But then there's another kind of cells, which are these green cells, which support these hematopoietic stem cells, and they support all the other tissues in our body. So we're starting to look at the potential of both hematopoietic stem cells, as well as what we call these mesenchymal stromal cells in terms of treatment for scleroderma. So for the stem cell treatment, and I understand that there will be a specific talk on this, so I'm not going to go into this in a lot of detail. But when we talk of hematopoietic stem cell transplant, what the idea here is that we believe that people with scleroderma have an unhealthy immune system. So what we do is we remove the healthy stem cells of this person that come from the bone marrow, and we put them away in a fridge somewhere. Then we give this person very, very, very strong chemotherapy to remove that unhealthy immune system. And when the person has no more of this unhealthy immune system, we give them their own stem cells back so that they can repopulate a healthy immune system. Does this work? It works. So there's been several studies, and I would say two very large international, they were national studies really, one done in Europe and one done in the US, that clearly show, and I'm not expecting you to understand everything, but that clearly show that both survival and organ involvement is protected in people who undergo transplant compared to scleroderma patients who do not undergo transplant. So these are the results of the ASTIS trial that were published 6 years ago, and these are the results of the American trial that were published in December 2018, which shows exactly the same thing. So if you remove an unhealthy immune system from somebody with scleroderma, you give them back their stem cells, these stem cells are able to repopulate a healthy immune system. And this really has a very important impact in terms of survival on our scleroderma patients. So here, focusing on the green, and what I'm trying to show you, this is European data. And so what I want to highlight is scleroderma is now the second leading indication for stem cell transplant in autoimmune diseases after multiple sclerosis. So multiple sclerosis is in blue and scleroderma is in green. And if you focus in down here, this is over time, and you can see over time there's really increasing use of stem cell transplant in scleroderma. This is European data. And so what we've decided, and I think the Europeans are ahead of us. And what we've started to think about in Canada is, well, if the Europeans are generating this really interesting data, I think the onus is on us to bring this treatment to our patients. So one of the first things that we did, and this is hot off the press, we published this about a month ago, is we said, well, if we compared European data, so this is patients who've been transplanted in France, to Canadian data, which are patients where we almost don't do transplant here in Canada, and we compare just their survival, we're seeing exactly the same thing. The European patients or the French patients who are undergoing transplant, they're surviving a lot better than the Canadian patients. And these are patients that have been matched to disease severity. So we are able to see that, that this is not only in clinical trials, but this is in real world practice as well. And if we sort of dig down a little bit deeper, it's not just survival, but the European or the French patients have a lot less pain. Their Raynaud's is a lot less severe. They have a lot less digital ulcers. It takes a while, but even their GI disease is a lot less important than the Canadian patients. Shortness of breath improves in the European patients post-transplant compared to our patients who are not transplanted. And overall disease is much, much better in the European patients. One of the things that we did notice, though, is that although quality of life, again, after transplant seems to be much better, so physical quality of life after transplant seems to be much better compared to scleroderma patients who are treated with routine care. One of the things that we've noticed, though, is that mental health-related quality of life really is not improved with transplant. And now we're trying to do some additional research in terms of trying to understand why is there this disconnect between the transplanted patients who are physically doing much better, and yet their mental quality of life does not improve by the same magnitude. So whenever you do a good research study, or the proof of a good research study is that it then leads to additional questions. So now we have to try to address these additional questions. So as far as I'm concerned, for hematopoietic stem cell transplant, there's definitely better survival, there's better improvement in physical quality of life, and this is sustained over many years. On the other hand, it's not associated with better health-related quality of, mental health-related quality of life, and we have to try to understand that. The problem with stem cell transplant is that very, very strong chemotherapy that is given to these patients at the time of transplant is very toxic, and the treatment itself is associated with a treatment-related mortality of 5 to 10%. So this is not something that we can do for every scleroderma patient. This is something that we can do for the most severe scleroderma patients. So I'd say about less than 5% of patients. It's worthwhile, but you have to meet the criteria. So what about the other 95% of the patients? What can we do for them? So just before I go into the next treatment, so we've developed this Canadian network for hematopoietic stem cell transplant, as I say, with the real rationale here of trying to now bring hematopoietic stem cell transplants for scleroderma as part of standard of care in Canada. If the Europeans have access to this and the results there are so much better, I think the onus is on your doctors to be able to say that you have access to the best care here in Canada as well. So lots of work ongoing in that field. So as I said, but the problem with stem cell transplant is it's really for the most severe patients, less than 5% of our patients. What about the other patients? So what I showed you before, which were these green cells that were supporting the hematopoietic stem cells, here they're shown in pink. And as I said, these are what we call mesenchymal stromal cells. They are in every tissue, and they really support all of the tissues. So here, for example, it's a blood vessel. And we know, for example, in scleroderma, that the blood vessels are not healthy. So we suspect that in scleroderma, these cells that are supposed to feed those blood vessels are not healthy. So there's a problem with the mesenchymal stromal cells in scleroderma. Can we replace these? And before I do that, so what do these mesenchymal stromal cells do? Well, they look at this. When I just look at a slide like this, these cells are supposed to be anti-fibrotic. They're supposed to feed your blood vessel. They're supposed to modulate your immune system. These are all the things that are going wrong in scleroderma. And so presumably the mesenchymal stromal cells in scleroderma are unhealthy. So how can we try to make them healthier? So where can we get these mesenchymal stromal cells? It turns out that we can get them in almost any tissue, but where we can get them in a most abundant quantity is generally from fat. So we can take them from the tummy fat. We can get them from the bone marrow, but that tends to be very invasive, very painful. But umbilical cord, so when babies are born, the umbilical cord is filled with mesenchymal stromal cells. And so people have started to look at the value of these MSCs to treat autoimmune diseases. And I can tell you that in Paris, with the collaborators that we're working with right now, they've used MSCs so far in 20 scleroderma patients, but they were getting their MSCs from the bone marrow. And as I mentioned, that tends to be quite painful and invasive. We're also working with some American colleagues who are actually using umbilical cord derived MSCs in lupus. So our idea was to try to combine the strengths of all of these groups and say, look, we're interested in scleroderma, but we don't want to use bone marrow derived MSCs. We're not interested in lupus, but we're interested in these cells. So what we did is we went ahead and we designed a study to look at umbilical cord MSCs in scleroderma. And we were lucky enough last year to be funded to do a large phase 1/2 trial, which is really an early trial, first of all, to see if these cells are safe in scleroderma, and also to start to see if there's a signal in terms of efficacy. So we call it the CARE SSC trial. We will be including 18 patients. It's a randomized placebo-controlled trial. So 6 patients will receive 2 doses of placebo. 6 patients will receive 1 dose of cells and 1 dose of placebo, and 6 patients will receive 2 doses of MSCs 3 months apart. And so we're in the process of trying to get Health Canada approval for this. And I think the good news is already this morning I received a follow-up letter from Health Canada, where they're looking at the fine print here, and they're trying to make sure that everything is okay to give us a go. But I'm hoping that we will have a go to start the trial at the latest at the end of this year or in early January. And our timeline is to get the trial done in the next about two and a half years. So hopefully we'll have some really promising results to present at some time in the next couple of years. So I think these are some of the broad strokes of our research program. I could talk about a little bit about the future and tell you that we have lots and lots and lots of projects going on. We have an interesting project, and so, we have lots of targets to do projects, because scleroderma is a disease that affects so many organs. So we're interested, as I mentioned, in the lungs. We've received funding to look at some newer biomarkers of lung disease, looking in particular at the proteome. In the kidneys, as I mentioned, we're in the process of doing the international scleroderma renal crisis study. Results will come in a couple of years. And perhaps what I will mention is one of the newer areas of interest that I think has been really neglected in scleroderma, and which has become one of our newer focus of interest, is the muscle disease in scleroderma. Understanding, what's going on at the muscular level, and how can we help our patients deal with this organ involvement. And so we've got all sorts of new projects looking at trying to identify new antibodies, understanding what's happening at the pathological level, the various mechanisms, and mostly trying to find the right treatments for our patients. So I will end there, and I usually end by thanking all of the people who have contributed to the CSRD and to the success of this group. There's many rheumatologists across the country. You might recognize your own. I also work with some scientists here at McGill. I work with people in France. But I think that, after having thanked all of these scientific collaborators, I think the most important person to thank is you, because I think that without patient participation and caregiver support and financial contributions from patient societies and so on, this research would really never have come to be. So I think that everybody who's listening today, I think, has a really big stake in this, and a lot of should take some ownership of the success of this work, because you've contributed to making it happen. So thank you. So
Thank you, Dr. Hudson. Sorry, yeah, we've got a question up. So it says, you mentioned that some research was showing early intervention helped improve outcomes. So why would stem cell transplant be better when it would be late intervention? So if you have a moment to share your thoughts.
Right. So first of all, cell transplant or hematopoietic stem cell transplant is also used in early disease, before the disease gets to be too late. So I think that the rationale is the same, both for lung disease. The earlier you treat, the better possibly the results you will get. The same is true with the cell therapies. The earlier you treat, and the reason for that is, for the most part, not always, but for the most part, most of the disease damage, and I showed that in one of the graphs, happens in the first 7 years of disease, and beyond that, a lot of the damage becomes irreversible. So cell therapies are also for the early phases of the disease, if you want.
That's the only question I'm seeing at the moment.
Okay.
I don't want to rush anyone in case there's someone's taking some time to type. Either way, I want to thank you, of course, for bringing us, we're so lucky to have you bring us such relevant and up-to-date information, especially when it comes to, studies like the one you mentioned coming out of Europe about stem cell transplantation, and that just being, only a month old. Those are such hot topics right now in our community, and so we appreciate that so much. So if there aren't any further questions, I'm going to turn my camera back on. If we don't have any further questions, I think we will. Oh, here we go. We do have another. Sorry. Okay, so Evelyn's asking, what part does stress play in your studies?
Yeah, I don't know that I have good scientific data to answer this question. What I do know is that the stress system in our body modulates directly our immune system. So to the extent that there's this interaction between the stress system and the immune system, I'm convinced that stress can impact immune function. The problem with that is that we can't we don't really control stress very well. That too we don't. And the other thing is, once stress has perhaps at the epigenetic level caused immune dysfunction, even though you take the stress away, the epigenetic dysregulation will persist. So yes, I think that there's an interaction between stress and immune function, but the consequences can be long lasting.
We have a question that says, what will the MSC do?
Okay, so the MSC, so as I showed you, the MSC have modulate the immune system. So that's the first thing that we're going to look at. So all of these cytokines and chemokines and inflammatory markers that are dysregulated in scleroderma, are those normalized? So that's one thing that we're going to look at. The second thing that we're going to look at is, what about the vascular markers, the markers that are, there's poor circulation in scleroderma. So can it rectify dysregulated angiogenesis, if you want? And then ultimately, we're also interested, because these MSCs are anti-fibrotic. So the efficacy outcome of the trial is actually going to be skin involvement. So the modified Rodnan skin score, looking at to see if they can somewhat reverse fibrosis. And I mentioned quickly that trial from Paris with 20 patients using bone marrow derived MSCs. And I have seen their data on skin scores at 3 months, and it is looking good.
Wonderful, thank you. Our next question, I see, what is happening in the SIBO-SABO pragmatic trial?
Okay, so the SIBO trial, the PI for that trial has been Dr. Murray Barron, who is here. He sits across the hall from me. I understand that the data has all been collected and that they're analyzing the data currently. So I can't answer that question. I don't have the result, but hopefully those results will be coming out soon.
Thank you. The next question, why can't I find information of juvenile scleroderma? Oh, you see there.
Yeah, oh, that's a very good question. So, you know, I don't see a lot of kids. First of all, I don't see a lot of kids. And secondly, scleroderma in children is extremely rare. And for the first time now, I've never even thought of the idea of having juvenile scleroderma, scleroderma. I have never come across any information on that. It must be very, very rare. I can't help you there.
Sorry, next, I see many of us have been living with scleroderma for more than 7 years. Of the research you discussed, is there any treatment being explored that is geared toward treatment of later stage scleroderma systemic?
Okay, so that's a great question. So there's a few things. So first of all, is active or progresses in the first 7 years. So that's why there's such a focus on those first 7 to 10 years, if you want. But, you know, I still see patients who have progressive lung disease beyond that 7 to 10 year time frame. So yes, in some of these patients with obviously progressive disease beyond the first few years, those patients are still eligible for some studies. The other thing, though, and I think more practically speaking, is that that's where things like, groups like SPIN come in, where the disease has reached its maximum, it has plateaued, it is no longer active, but the patient is left with all sorts of damage. That's where, you know, there's a lot of research going on for people with established disease, trying mostly addressing issues of rehab and psychosocial interventions and so on.
Our last question comes from, oh, well, maybe not our last. It comes from Jillian. Will MSC treatment also be indicated for early in the disease?
Yeah, so I think that they're best indicated for early disease. So as I said, that's where I think we can probably have our biggest impact. So yes, they are in the study that we're going to be conducting, we will include patients with early disease. We will also, however, I don't think that disease duration is a criteria. So we will take patients with established disease as well.
I see Mark has shared a question with us. Have you worked with Dr. Torek in Pittsburgh? I know Mark's daughter is I can see everyone can see there, she's 10 years old. And if you'd want to address that question.
I think that's a great comment. I have never worked with her. I know she's very well respected in the field of juvenile scleroderma. And in fact, if anybody knows about scleroderma, scleroderma, I would think that she would be the right person to speak to. And having seen her present at meetings, she looks like a really very nice and approachable person. So I'm sure that she would be willing to take on questions.
That's great, thank you. So at the moment, and I think we're keeping in time, that it looks as if you've answered all those questions. Thank you. that wrapped out just well, that was just perfectly, thank you. Okay, so, you know, obviously, again, I can't thank you enough for being with us today. The work that you do for the scleroderma community is so impressive and it's so incredible. And I think I speak for all of us when I say thank you for what you do and certainly for your time today.
Thank you. Thank you for
having me.
Okay.
Have a great day.
Thank you.